All right, so we're going to be talking about DNA and its role in heredity, how we discovered that DNA is what contains our genetic information, and the structure of DNA itself. So early experiments that explored the question, what is the genetic material, were performed by Frederick Griffith, Avery McLeod and McCarty, and Hershey and Chase. Frederick Griffith used Streptococcus pneumoniae, which is a form of bacteria, and it has two types. It has a smooth colony with a capsule around it that causes pneumonia, and a rough colony which does not have a capsule around it, and it doesn't cause pneumonia. So if it doesn't cause it, we call it avirulent. If it causes something, we call it virulent. And then, so in his experiment, he took virulent smooth colonies and put them into the mouse. And when he did that, the mouse died. When he took the avirulent rough colonies and injected it, the mouse lived. Then he heat killed the S strain and discovered the mouse lived. So when he then incubated the heat killed S with the live R colonies, the mouse ended up dying. That told him that something was being transferred from the heat killed S strain into the live R strain. He didn't know what that was, but he knew it had to be the factors that Mendel was talking about. We didn't know specifically what those factors were at this point, but he knew it had to be those factors. This whole process we call the transforming principle, and that's when you can use one molecule to transform another molecule. So then in 1944, Avery McLeod and McCarty decided to figure out, well, what was that transforming material? What was being transferred from the smooth to the rough? But they took care this time, using the same test as Griffith, to exclude both protein and RNA as a chemical agent, and discovered that DNA was the chemical agent of transformation of the S pneumonia from avirulent to virulent. So they went off the theory. After their experiment, they concluded that DNA was a genetic material. However, in the 20th century, scientists still debated and doubted that DNA was the genetic material because it only consists of 4 bases that repeat, and they couldn't see how that could contain the amount of information that codes for an individual. So Hershey and Chase in 1952 set off to prove that. And they demonstrated an experiment called bacteriophage infection. And using that experiment, they concluded that DNA was the genetic material, not proteins. So this shows you the Hershey and Chase experiment. So these little alien structures are called bacteriophages, and they are viruses that infect bacteria. And so what they did was they took one bacteriophage and identified or radioactive labeled the protein in it, and then they took another bacteriophage and radioactively labeled the DNA in it, allowed them to infect a bacterial and replicate using the bacterial's cellular metabolism, upon which they discovered that the only one that ended up being radioactive was the one with the DNA. And so that proved that DNA was the genetic material, not proteins. So the classic molecular biology experiments performed by Griffith, Avery, McLeod, and McCarty, Hershey, and Chase combined revealed that DNA is our genetic element. Then you had a woman named Rosalind Franklin in 1951, and she used X-ray crystallography to determine the positions of the atoms of DNA, and that the pattern of DNA is both highly regular and repetitive. She collaborated with a man named Wilkins. You can see here, this is an actual picture of, or X-ray crystallography image of DNA. And if you act as if you're looking down into a tunnel in it, you can see the circular or helical structure of it, and the repetitiveness of it. So her experiment helped us to understand the structure or chemical composition of DNA. So biochemists knew that DNA consisted of nucleotides, and that those nucleotides consisted of a deoxyribose sugar, a phosphate group, and a nitrogenous base. There are 4 bases: adenine, guanine, cytosine, and thymine. Adenine and guanine are what we call purines, and cytosine and thymine are what we call pyrimidines. Now, in 1950, Erwin Chargaff found that the amount of adenine is always equal to the amount of thymine, and the amount of guanine is always equal to the amount of cytosine, stating that a purine always bonds with a pyrimidine. Never have a purine to a purine, and you don't have a pyrimidine to a pyrimidine. So that adenine always bonds with thymine, and guanine always bonds with cytosine. So you can put apples on the tree, gas in the car. It's kind of how I remember it. The 3D model building structure of DNA came from Francis Crick and James Watson, using Franklin's X-ray crystallography that helped convince them that the molecule was helical. So they used density measurements, which suggested that there were 2 polynucleotide chains in the molecule, and that the modeling showed that the DNA strands had to be anti-parallel. There's Watson and Crick looking at their model. So Watson and Crick suggested that the bases, the nitrogenous bases, adenine, thymine, guanine, and cytosine, were on the interior of the helical structure, and the sugar and phosphate were the backbones on the outside, not the opposite, with the bases on the outside. So they call these 2 strands anti-parallel. That is, the complementary strands run opposite of each other, or they're flipped of each other. So in the sugar phosphate backbone, the phosphate groups are bonded to the 5 carbon of one sugar and the 3 carbon of the next. So if you look here, you can see the ring structure of the carbon of the deoxyribose sugar. And each carbon we count, we number it: 1, 2, 3, 4, 5, 6. Okay. Oh wait, sorry, 5. That's an oxygen, not a carbon. And so what they discovered was that the phosphate groups bond to the sugars at the 5' and 3' carbons. And you can see that over here, where you've got the 5' and the 3' with the phosphate groups attached to it. Okay. And again, this phosphate attaches to this 5', and then the 3' would attach to another phosphate. So we identify the strands with the 5' facing up as the 5' end, and the 3' on the bottom as the 3' end. So we found that the strands are actually, they consist of 2 strands running in opposite directions that are held together by base pairing between purines and pyrimidines opposite one another on the 2 strands. So you can see when you're looking at a strand, one side will have the 3' carbon up, facing up. The other strand will have the 5' carbon facing up. So they're kind of flipped of each other. The 2 anti-parallel strands in the DNA molecule are twisted into a double helix. Per Chargaff's rule, a purine on one strand is paired with a pyrimidine on the other, making the base pairs adenine, thymine, and guanine and cytosine the same width down the helix. Okay? So they're structured as such, not this way. So you end up with this funky structure. You get an even width of the bases down the helix. So your purines are adenine, guanine, and your pyrimidines are thymine and cytosine. Is A to T, which consists of 2 hydrogen bonds, guanine and cytosine, which consists of 3 hydrogen bonds. 4 key features of the DNA structure. The double-stranded helix of uniform diameter. The chains are held together by hydrogen bonds, and then between the base pairs, and then by van der Waals forces between adjacent base pairs on the same strand. So here you can see you're showing the hydrogen bonds are what hold the bases together, and the sugar and phosphates are held together by covalent phosphodiester bonds. And that they are anti-parallel, 3', 5'. The outer edges of the nitrogen spaces are exposed in what we call major and minor grooves. The grooves exist because the helices are not evenly spaced. Or the twists is what they're saying. There are 4 possible configurations of the flat hydrogen bond base pairs with major and minor grooves. So if it bonds this way with guanine on the left and cytosine on the right, and adenine on the left and thymine on the right, you have your major grooves up here and your minor grooves down here on the opposite side. If it bonds this way with cytosine first and guanine, and then thymine and adenine. Okay, so those are the 4 possible configurations of the base pairs within the double helix. The atoms shaded in the purple are available for hydrogen bonding with other molecules. And these differences in the configuration allow proteins to recognize specific DNA sequences. And then this just shows you here's a prokaryotic chromosome. It's one circular chromosome. And here's a eukaryotic chromosome. They contain multiple linear chromosomes. Prokaryotics contain a single circular chromosomes. And then there's a list of information that you can read on your own. So that's DNA, how DNA came to be understood as containing the genetic information of organisms, as well as the basic structure of DNA. Our next video is going to talk about how DNA actually replicates itself. Have a good evening.